Hey everybody, welcome. This is the Rotary E-Club of Silicon Valley, and every week we bring you the stories of those who make the world better locally, globally, and digitally. We're part of Rotary International, 1.4 million Rotarians and Rotaractors. There are 36,000 Rotary clubs around the world. We're all groups of people who are dedicated to service above self. That thought that there are things we can do with our time, talent, and treasure to make the lives of others better. These stories that we share as a part of our club's online and asynchronous format, which we have been doing for over 10 years. We've got 500 of these recordings or so just to make you go, man, the world's a great place, are ones that are dedicated to giving you some hope for the future. In terms of making people's lives better and giving hope for the future, boy, do we have a program lined up for you today. With us is Dr. Matthew O'Connor. And Dr. Matthew O'Connor is somebody that I think is going to give us a message that is really, really exciting. He is the former vice president of research at SENS Research Foundation, now the Lifespan Research Institute, and has overseen all kinds of projects related to rejuvenation biotechnology, which we'll need to discuss, like the difference between that and something that seems a little more science fiction, right? And especially the research that you are doing and the trials related to atherosclerosis, which I hope I have pronounced properly. We'll see. All right. But as one of the founders of Cyclarity, he's going to talk about the work that his company is doing. And Dr. O'Connor, it is an honor to have you with us. We are excited to hear your presentation, and I hand the mic over to you.
Thanks so much, Rushton. It's an honor and a pleasure to be here. And the you were talking just now about the Rotary Club, the E Club of Silicon Valley, and the Rotary Club International, and we were talking about this a couple of weeks ago when we set this up, and it really resonated. Your dedication to service really resonates with me. We're a private company, but you mentioned my time at the foundation, which was an aging research foundation, and it everybody who works in my company is completely mission driven. And we formed this company as a spin out of a nonprofit in order to save lives. And of course, everyone wants to make $1 billion, for the people who invented it and for the investors. But what I said to my team last at our holiday party last year was, sure, it would be great to make $1 billion, but I don't think we're going to be real billionaires until we save 1 billion lives. And that's the kind of billionaire that that I want to become. Uh, so you mentioned the oxidized cholesterol. Let me take a slight step back from that and sort of talk philosophically, and we can talk more about it and about rejuvenation and what that means later. But the basic idea, my philosophy of approaching chronic disease, which is the major killer, all the major killers in the world today, the biggest killers all revolve around chronic disease, and the biggest risk factor for chronic disease is aging. And what I like to think about as an approach to tackling chronic disease is, what is it about the damage that accumulates with age that we can remediate, that we can remove? And one of those things is oxidized cholesterol. Oxidized cholesterol is what happens when you have regular cholesterol that gets reacted with an oxygen-free radical, and it just adds one oxygen atom to it, and boom, you have something that's very toxic and completely useless. Your body can't use it anymore, and it doesn't have good ways to process it, to metabolize it. So I've actually been thinking about this particular problem for about 20 years, since I was in graduate school, and I saw a scientist and mentor of mine, actually the founder of the SENS Foundation, give a lecture about this at my university. And so I've been thinking about this for 20 years, and it was about 10 years ago that I actually started to do something about it when I was at the foundation. And and so the idea is that this, wherever you have cholesterol, you're going to have oxidation, and it happens in the brain and it happens in the blood vessels, and that's part of what contributes to atherosclerosis. This has been a known problem since the 1990s, but there's been a ton of research since then. Now a couple hundred studies about this problem, and finally, in the last 10 years, some good clinical data. So this stuff that I'm showing here now shows the association of levels of oxidized cholesterol. And by the way, 7-KC stands for 7-ketocholesterol, which is the major oxidation product of cholesterol that happens when you have that random reaction. It's not completely random. It basically tends to be just this. And so um, what we see is a strong association between the buildup of oxidized cholesterol and disease severity. So how does this happen? What happens in the buildup of plaque in your blood vessels, a process that's called atherosclerosis, is that you get a small lesion in your blood vessel wall. You can get a little lipid deposit, and then an immune cell called a macrophage is supposed to come along and eat that up and repair it, and then go away. But what happens when you get a big lesion that builds up and turns into a big plaque is that you get these things called foam cells, which are macrophages that have become too engorged with lipid, and now they just look like big blobs. That back in the 1950s, all we had was a microscope picture of these big blobby cells inside of these plaques, and so somebody named them foam cells. What happens there, we believe, is that when they eat regular lipids, like they're fantastic at processing them, but when they run into oxidized cholesterol, they get stuck. And they can't process lipid anymore. They can't metabolize the oxidized cholesterol, and they stop doing metabolism, and they just keep eating and ballooning up into these blob cells. And so what we and other groups in recent years have thought is there a way to supercharge the macrophage and put them back to work clearing up the plaque? And from our perspective, we decided that the best way to do that was to remove the root cause, which was the oxidized cholesterol. So, like I started to say earlier, about 10 years ago, I was thinking about this problem, and I went back into the scientific literature from the 80s and 90s and found these molecules called cyclodextrins. Cyclodextrins are circular carbohydrates that are produced by microbes naturally, and they have all kinds of interesting applications. Technologically, they're really engineerable, and they have all kinds of industrial medical applications. In medicine, they've been used to deliver drugs, and more recently, the first drug itself that's based on a cyclodextrin came out, which is called Bridion, and it's owned by Merck, and it outsells the rest of anesthesia combined. It's such an amazing drug as an antidote for an anesthesia agent. So what we're doing is the same kind of thing as that Bridion drug does, which is try to sequester and grab the target, which in this case is this yellow oxidized cholesterol in the middle of this these cyclodextrin molecules. And so there's two cyclodextrin molecules here surrounding this. And what we've done is we've dimerized them. We've stuck two cyclodextrins together, and that very, very simply is the crux of our innovation, is that we've stuck two cyclodextrins together in the right configuration, engineered them the right way so that they can grab onto the oxidized cholesterol really strongly and specifically. So there's only one atom difference between cholesterol and this oxidized cholesterol, and we trained this molecule to strongly prefer the oxidized cholesterol version. And that's the secret to the how our drug works and and why it's safe. Because actually, if you have something that binds cholesterol too strongly and can pull it out of cells, out of cell membranes, it can kill cells, and that can cause toxicity. And so we discovered early on that that was the secret to making our drugs safe, was to make them not like cholesterol. So, just some really brief scientific data. I'm not going to go too deep into the science, but if you want to go deeper later on, we can. But th this is kind of some of the most important things that I have here, which is that we can grow some healthy macrophages. It might be hard for you and your viewers to see them, but there's a bunch of these small macrophages on the plate. And then when we give them just a little bit of oxidized cholesterol, they quickly turn into these foam cells, which here are portrayed with this staining of lipid that we did. So they sequester a lot of lipid, these things called lipid droplets, and that's the hallmark of a foam cell. And then for the first time ever, I believe, and we're going to get to publish a pretty cool paper on this because of this phenomenon. We've reversed that and turned them back into healthy, normal looking and acting macrophages. So they go back to doing the job of healthy young macrophages. And then over here on the right, this is kind of a bit of a ugly graph, but the key to this is, number one, I'm not showing the data, but our drug acts really fast. It acts within 15 minutes. It gets rid of the oxidized cholesterol that fast. It doesn't mean that it's going to make you feel better and clear out your plaque. It's not Drano. It's not, it's not flushing out the pipes like that, but it acts at the molecular level within 15 minutes. And part of the safety trick of our drug is that it's cleared from the body within hours, not days. It has a short half-life, and so you're going to pee it out in a couple of hours. And what we see in rodents is that not only are they peeing out our drug, but they're also peeing out oxidized cholesterol, and they do it really fast, and they do it in time that mirrors when our drug is coming out. So everything is working as designed, and this test, this really sensitive urine test, is going to help us a lot in our clinical trials. So just some brief information about our clinical trial plan and status. We're running our first clinical trial in Australia, and we're running it under the supervision of the world famous cardiologist, Dr. Stephen Nichols. And he is the world's foremost expert on plaque and how it's formed. And he has overseen many clinical trials for the cardiovascular drugs that people take commonly these days. So we have this phase 1 clinical trial. It's going into 72 healthy volunteers to make sure it's safe. And we're also going to get to see some data like the urine data from the rats in these healthy volunteers, because our test is so sensitive that it should work on even the young healthy volunteers, not just on the older sick patients. We started this trial in January. We're through the second cohort now, and going through 6 cohorts, and then doing it a second time in a single dose and then multiple dose. And then later this year, we'll get to go into patients, and the patients will have plaque in their coronary arteries. And we'll do some imaging of the plaque before and after our treatment. And so we'll not only get the data showing that we're removing the toxic oxidized cholesterol, which we'll get very fast, but later we'll also be able to get a picture of their arteries. Only 12 patients, but that will then expand next year into a phase 2 trial where we'll go into a lot of those patients. So that'll basically be a scaled up version of that tiny little pilot that we're going to do later this year, where we'll test on about 150 patients with that same disease and see if we can see some nice robust data on them, both on the direct effect of the drug removing the oxidized cholesterol and hopefully making the patients feel better and hopefully clearing out their plaque. And that's the holy grail of our approach and I think of the field generally, because there's no way right now to reverse plaque once it's there. Right now, basically, all you go to your cardiologist and they say you've got plaque. Everything that they're going to tell you to do, they're going to tell you to quit smoking and exercise more and eat a better diet, and they might put you on a lipid lowering drug like statins. None of that is going to reverse your plaque. It's going to slow it down, but it's not going to reverse it. So that's what we're trying to do. Some people, the leadership of our team, me and Mike, my co-founder, started the company. Noah Rosenberg is our head of clinical, and he put together this amazing clinical advisory board of world famous cardiologists. I already mentioned Dr. Steve Nichols. He's down in Australia, the head of the Victorian Heart Institute and the new Monash Heart Hospital, which is just an amazing state-of-the-art heart hospital that I got to tour last spring, and PK Shaw, who discovered the process of cholesterol oxidation in blood vessel walls, and Mike Farku, who's the head of research at Cedars-Sinai, and ran some really amazing, groundbreaking clinical trials in cardiovascular disease. So we're targeting atherosclerosis, the plaque, first with our drug, but since this is sort of a basic first principle of biology that oxidized cholesterol is toxic, these are other diseases that oxidized cholesterol is implicated in, and things that some of them we're just starting to work on, others that we hope to work on in the future, and there's a lot of potential there. And if there are people in the audience who want to collaborate on any of these other diseases, please reach out. I'll just briefly touch on our basic platform before I wrap up, which is that I haven't really talked, and I don't have time to get into it too much, but we have this computational chemistry platform that we used to invent this first drug, and that we're now using to invent the next 10 drugs that we're going to produce to target many other toxic molecules that accumulate, some that accumulate with age, other that are environmental. And we've just now added on machine learning and artificial intelligence to the point where we're taking the human out of the process of designing the drug. So previously, we were designing the drug by hand and then plugging into the computer, running a simulation, seeing how the numbers look, and then based on that, making a guess about what might work better. Now we're just letting the AI iterate by itself and do 100,000 or a million simulations and have it spit out the best candidate for us to build. These are some things that we're working on. Some of them age-related chronic disease, others less so, like anesthesia reversal, which like antidotes, like I mentioned before, for Bridion, which is a nice test case and a perfect application for our technology. Macular degeneration. Something we haven't really started working on yet is like poison ivy rashes, but that thing, Urushi oil, which causes the rash from poison ivy, is a perfect target for our drug. And then nanoplastics. A lot of people are worried about that right now. Those are also a good potential application for our technology. And there's other aspects in other non-therapeutic areas like food science, material science, environmental remediation that could be good applications that we're not really experts on. But again, we invite people to come and collaborate with us. So that's what I have to present today, and I can stop there and take questions.
Fantastic. Okay, I am excited to have our group be able to dive in on this. Let me introduce the people that we've got so far. Members of our club, first of all, our president this year in the Sacramento area, Mike Kane. Here in the San Francisco Bay Area, chess evangelist, Father Deacon Christopher Major. In the Houston area, Rory Olson and sidekick Gus. So I am Rushton Hurley. I'm loaded with questions for this. We're going to start with one that both I and Mike had looking into this is that in thinking about addressing these issues, there are all kinds of things that I think people hear about the idea of how to kind of prevent oxidized cholesterol in the first place. So let's get kind of a lifestyle idea out of the way to begin with. What are the kinds of things that people, I mean, should have clearly in mind? These are the things that lead to oxidized cholesterol buildup. Is it fatty foods, fried foods, polysaturated fats? I mean, what are the kinds of things that lead to needing the kind of treatment that your research is looking to address?
Yeah. On one hand, I want to say, somewhat depressingly, that it's a somewhat inevitable process. Like everybody has cholesterol in their bodies, and oxidation isn't something that you can completely do away with, and even regulating it is tricky. Having elevated cholesterol levels is definitely an issue, and for that eating a diet high in saturated fats, and especially probably highly fried foods, things like that are some of the worst ones. Um, I, Ironically, eating oxidized cholesterol when you have a young, healthy liver isn't it doesn't seem to be at all dangerous. But something that hasn't been sufficiently studied and isn't regulated at all by organizations like FDA or USDA is eating oxidized cholesterol when you're older and or if you have liver failure. And in animal models, it has been shown that animals that are undergoing liver failure are having problems eating oxidized cholesterol. So, the cholesterol in your food, like you were saying, like if it's being fried and getting oxidized that way, or if it's been exposed to very high heats in the presence of oxygen while it was being prepared, then you could be eating a lot of oxidized cholesterol, and that could damage your liver. But like as I said, it's not well studied enough, and it's not something that's regulated at all.
So following on that, the kinds of things that become issues for people, is oxidized cholesterol a factor in the development of Alzheimer's disease?
Yes, yes, it is, and it's something that we actually got a grant from the National Institute on Aging to study our drug to target the the oxidized cholesterol in Alzheimer's disease, and it has been implicated in Alzheimer's disease and other forms of dementia, and so. When the brain is the highest concentration of cholesterol in your body, and when you start going down that path of brain aging and dementia, it creates a somewhat hyperoxidized environment, and that does contribute to Alzheimer's disease and other dementia, probably by damaging the macrophages of the brain, which are called microglia. That's what I think is going on there, and how we might in the future be able to start to address that.
Okay. And as you do these trials, and they've started in Victoria, and is this part of the raft of things you're measuring to try to get a sense of where the drug that Cyclarity has developed, might be you might need to essentially focus your time as you begin to develop kind of practical outcomes of the research?
Yeah, we're you always want to focus a lot in your clinical trials on, one disease indication and really try to measure that. And so mostly we're focused on, first on safety, second on whether or not we can discover some early efficacy, and then really focus in on the efficacy in the later stage clinical trials. But I think there's a decent chance that we're going to start getting some observations of people who have multiple feeling older and sicker and feeling having a bit of starting to have some undiagnosed cloudiness in their brains, having slowed down a little bit. I think we're going to start to see some people as we go on with the patients who are going to start feeling more vigorous, who are going to be able to have more energy and be able to move around better. They're going to be breathing better and maybe even thinking more clearly. And then we'll be able to, and once we start getting some of those observations, then we'll be able to design some clinical trials focused on some of those things. Just one at a time.
Cool. Speaking of the trials, you've got this partnership with the Victorian Health Hospital in Australia. Do you have other trial locations that are set to move forward in the coming years that you can talk about?
Yeah, we're working on going international in our phase 2 trials. So we're hoping to bring it to the US. We're talking to Cedars-Sinai. We're looking at other countries. We're looking at the UK. We actually won an accelerated regulatory award in the UK. So we're looking at doing trials there. We're looking at Europe and the European Union and South America. So we're going to try to expand to as many places as we can, as is practical, and get people from many different backgrounds on our drug. Absolutely, we're going to try to do it as far as we can, and then in the phase 3, it'll probably be even further.
So in gathering data on these things, is there a current test that is common all over the world that measures oxidized cholesterol buildup? This is a question that came through the chat. Like, are you able to draw on public data that allows you to have a sense of normal levels for different age groups in specific populations or specific geographies or anything like that?
Good question. Not enough. There's not a commercial test for the oxidized the main oxidized form of cholesterol that I'm talking about. So it's really hard for for people to go and get a diagnostic test to see if they might be a better candidate for a drug. We have a test that we've developed. It's relatively expensive, and it's right now we're not looking at commercializing it. We're hoping that others will in the diagnostic space will come along and come up with a good cost-effective way to develop such a diagnostic test. There is, savvy listeners may say, wait a minute, there's a test for ox-LDL, and that's true. And so that's kind of a surrogate related test, and it's a little bit complicated why they're not the same thing. But LDL and HDL are actually not cholesterol. They're giant masses of particles that contain cholesterol, that transport cholesterol, and you can measure these giant particles when they're oxidized, and they are carrying oxidized cholesterol. So that's a surrogate test that people can get that isn't necessarily a one-to-one for what we're doing, but it's pretty close, and it's the closest we have right now.
So I'd like to build off another question that came in, and you talk about testing these drugs. Obviously, a kind of proper test includes a placebo, and is it the case that there are kind of common natural ways to lower cholesterol that you have to screen for as a part of your research so that you don't end up with a bunch of people taking the drug that aren't doing this thing that kind of everybody should be doing, and you end up with a placebo group that is living healthier in some fashion, that make it more difficult for you to measure the effectiveness of the drug.
Well, yeah, three things. One, of course, our trials are all placebo controlled. Even at the basic safety level that we're at now. Two, um, there's a lot of what we call exclusion and inclusion criteria, both for healthy volunteers and for patients. So we try to control for a lot of those things. There's a lot of substances, like right now, the people, being tested with our drug they can't be smokers. They can't be on any medications. And they can't even be drinking coffee. Like that's how seriously like we take and clinical trials, well-run clinical trials generally try to remove those confounding factors is by really simplifying it and trying to make everyone's lifestyles as close to each other's as possible. Then when you get into the patient side, you have people who have to be on standard of care. So, it may be that they're all going to be on a statin to keep their cholesterol lower, and hopefully, they're under the supervision of a cardiologist who's encouraging them to eat well and exercise well. But of course, there's going to be variability in there, and that's where, sure, you're going to have more variability in a tiny group like 12 people that we're going to look at this year, less next year when we're looking at 150 people, and even less in a couple of years when we're looking at maybe 1,000 or even a few thousand patients later on when we get into the phase 3 trials.
Well, it's particularly exciting stuff, and I love that the point of departure is to, I think I'm quoting you right from the slide, save a billion lives by targeting the root causes of disease. That's
Absolutely. Yep.
that's a heck of a frame from which to go, and I can imagine why the people at Cyclarity are mission driven, in that regard. I'm going to wind things down and then hand it back to you for the final word. For all of you who have joined us, thank you for taking your time to spend with us. We believe that the people who present to us are doing important things, that they not only have this potential to improve communities, but to inspire us to remember that there are loads of people out there who are making the world better. The news does not always reflect that reality from our perspective, and we hope that you will come back again and again and enjoy the kinds of things that we get to share. If you are interested in Rotary, go to rotary.org. You can learn all about this organization that is all over the planet, and you can learn specifically about our club by going to rotary.cool. So with that, we always like to hand it back to our speaker for the final word. Dr. O'Connor, what would you like people to have clearly in mind as they finish watching or listening to the recording?
Well, I thank you for your interest and thanks so much for the opportunity today. And I encourage people to follow us to get educated about cardiovascular disease and ways in which the aging and then a damage repair approach to remediating the diseases of aging, chronic disease. Look us up on our website, cyclaritytx.com. Follow us on social media, and reach out. I may not be able to help everyone who writes, who's having, health problems right now, but we love to hear from you. And if you want to collaborate, if you're in research or in pharmaceuticals and you want to collaborate or you just want to talk, definitely reach out. Investors contact us, and we're happy to talk to everyone. So thanks so much for having me here today.
Well, it is our pleasure, and we thank all of you who have watched this, and we will see you next week.